Hello, everyone, and welcome. I'm Professor Angharad Davis. I'm a consultant medical microbiologist by background, based at Swansea University Medical School and the North Wales Medical School. And I'm also Clinical Director for Publishing and Engagement at the Royal College of Pathologists. Thank you very much for joining our webinar tonight. I'm really delighted to welcome you. I'll be handing over shortly to our webinar hosts, who are Zainab and Rosie, but I'm just going to start by introducing the Royal College of Pathologists and giving you a little bit of background. We are a professional membership organisation with over 12,000 members, mainly pathologists and clinical scientists working in 17 different pathology specialties. Most of our members work in hospitals and universities in the UK, although around a quarter of our members are international and work and train in a wide range of countries. And an important part of the college's work is to oversee training, exams and professional standards across the pathology specialties. We also have undergraduate membership and a foundation fellow scheme for foundation doctors. Undergraduate membership is free, and if you're in a pathology specialty for foundation, you can also apply to be a foundation fellow. So take a look at the RCPath website or get in touch if you'd like more information about that. Our speaker tonight, Dr. Matt Clark, who will be formally introduced to you in a minute, is the college's undergraduate and foundation lead. And his role involves leading and coordinating the college's undergraduate and foundation activities to raise awareness of the pathology specialties and inspire the next generation of our pathologists and clinical scientists. So I'm now very pleased to be able to hand you over to our medical student hosts, Rosie and Zainab. Thank you both.
Thank you for that lovely introduction. I'll just briefly introduce myself. So my name's Rosie. I'm an undergraduate medical student, and I'm also currently chair of the Manchester University Pathology Society. And Zainab, if you'd like to just introduce yourself as well.
Yeah, thanks, Rosie. So hi, everyone. I'm Zainab. I'm a fourth year medical student and I'm pathology chair at UCL. So Rosie's just going to introduce Dr. Clark to you.
We have been supporting the college in running a series of careers webinars over the past couple of years. We're delighted that Dr. Clark has agreed to speak to us about the advances in genomic testing and how this is helping to improve the accuracy of diagnoses in pathology as well. As well as his role with the college, Dr. Clark is a specialty registrar and National Institute for Health and Care Research clinical lecturer in diagnostic neuropathology, who's also based at Great Ormond Street Hospital for Children for his clinical work, and the Institute of Cancer Research for his research. Dr. Clark will speak for 20 to 30 minutes, and this will be followed by a Q&A, which Dr. Clark and Professor Davies will answer your questions. So please just make sure to put your questions in the Q&A box, and we'll answer them at the end. So I'll now hand over to Dr. Clark.
Thanks very much, Rosie. I'll just share my screen. Okay, everyone, hopefully you can all see that. So good evening, and thank you so much for joining, and thank you very much for the kind introduction. So my name's Matt. I'm a neuropathology trainee, just coming towards the end of my training, and it's a pleasure to be with you here tonight to talk to you a little bit about genomics, the value of reading the genetic code. Just to give you a little bit of background about this, I don't profess to be an expert in genomics at all, but what I have done is throughout my training, I've been using genomics in neuropathology almost every day. I'm in clinical practice, and also having studied it as part of my PhD, which I'll tell you a little bit about shortly. This has meant that I've developed a bit of background, a bit of experience related to it. When I was at your stage as undergraduates and as a junior doctor, genetics and genomics used to fill me with fear. I always struggled with it at medical school. I found it really interesting, but really struggled to get some of the concepts associated with it. But hopefully, from listening to the talk that I give tonight, we'll go through some of the basics associated with different genetic tests that we do and highlight to you some of the value that these have in our day-to-day clinical work. And so hopefully for some of you who may have been feeling similar to I did back at your stage, this may encourage you to give it another go and have another look at it and explore it a bit further. So I hope you'll find it useful and interesting from whichever perspective you're coming at it. So first of all, just to introduce this, I just wanted to show you this picture, which is quite an old picture now, known as the Tree of Medicine. And I really like this picture because it showcases the real value of pathology. The branches of the tree represent all the different clinical specialties, and such as surgery, psychiatry, dentistry, neurology, internal medicine, and pediatrics. So they are all the branches. But it is general pathology, which is the trunk holding all of these branches up. And then the root system is down the bottom here, representing the different basic sciences, including the different pathology specialties. So they have an integral role in ensuring the proper functioning of the clinical specialties in a day-to-day running of the healthcare system across the world. And one of these roots is genetics or genomics. And I would also argue, and perhaps you will see as a result of this talk, that root actually has a lot of links to the existing root system as well, and it's vital now in our day-to-day work for helping our patients. The other thing to talk about is the traditional approach to treating patients. And it used to be that we would treat patients who had the same disease in exactly the same way. That used to be the general protocol. But now, in the more modern age, we're looking at a concept of personalized medicine, where every single person that comes in with a particular diagnosis or a disease, even if it's the same, will be treated in a different way. And part of the reason for those differences in the way they are treated is because of the underlying genetic changes associated with their diseases. And we now know that even patients that have the same disease may have different molecular or genetic characteristics that make them slightly different. And so the genomics is actually helping us to refine how we make a diagnosis, but also how we treat disease as well. The other thing to promote about genomics is it's a very cutting edge and very fast paced specialty. And there are numerous different tests and techniques that we have available to us to help achieve a diagnosis or refine that diagnosis for our patients. Each year, there are new developments in the genomics field. So there may be new discoveries related to genetic changes, but there also may be new techniques that we can utilize as well, including different sequencing modalities and also things like fluorescence in situ hybridization or immunofluorescence. And one of the other nice things about it is you often get a lot of pretty pictures associated with these results to look at, which is always very satisfying as well. Something that's a very modern concept now is known as the integrated diagnosis. And years ago, when I first started my histopathology training, we would get cases, we would look down the microscope at the sample, we'd have a bit of clinical information and history associated with the patient. I'd look at the cells, look at some of the features of them and their relationship to each other in the tissues, perhaps do a few immunohistochemistry stains, and then I would write a report giving a diagnosis that would then be signed out, and that was the end of the story. Things have significantly moved on now, and that is because of the genomics revolution that has brought a whole range of different other investigations that we have. And each one of those brings has its own value and represents another jigsaw piece in helping us to produce an integrated diagnosis. And that also goes alongside things like digital pathology and also now on the horizon, the use of artificial intelligence. So all of these different components now are forming the integrated approach to bringing a diagnosis together, and it's the pathologist that is actually leading that process. So making a diagnosis now consists of many different processes, one of which is included in many different forms of genomics testing. So who are the people that are actually involved in genomics testing? Well, there are a huge variety of people that are involved, and that goes right the way to the patient themselves and also those clinical teams on the ward who are looking after that patient as well. And whether that's the medical team or the surgical team, we may receive a fluid sample or a biopsy specimen or a surgical resection, which are then processed within the lab by the biomedical scientist team to give us our slides that we can then look at using the microscope or the digital screen. And the pathologists can then make an assessment of what they see microscopically, either using a microscope or the digital screen, as to what diagnosis they think they're dealing with here, and then decide whether molecular testing is going to be valuable in these particular cases, but also then marking out on the slides which are the areas of a particular tumor or disease process that might be worth exploring more about the genomic characteristics of. We also work extremely closely with our extremely valued clinical scientist team who have the huge responsibility of processing these tissue samples, i.e. extracting the genetic information, whether that's DNA, protein, or RNA from the tissues, and then preparing it for sequencing at a process known as library preparation, and then undertaking the actual sequencing processes itself. And they're often dealing with tiny volumes of fluid, which contain huge volumes of data at the end after the processing. And they are involved then in the analysis of this huge volume of data output as well. And alongside and working with them, we have our bioinformatician teams or our data analysts. And again, it's their responsibility, along with the clinical scientists, to align the different genetic test results and provide the pathologists with particular conclusions that have been drawn from these reports. And then the pathologists will then try and integrate all of that together, look at those results and decide, is there anything that's going to make a big difference to the patient's outcome here? Is there anything that's particularly targetable, that we have a drug available that we can use? And then this is put together in their report, which goes back to the clinical team on the ward and ultimately the patient as well, and brought all together as part of the multidisciplinary team. So this is a very simplified version of the personnel involved in this. There are numerous other people that could be mentioned as well, but the important message is that there is a wide variety of different roles that lead and help to support genomic testing in pathology. But why is it important, and why have we seen this sort of revolution of the input of genomics within medicine and pathology? Well, as I sort of alluded to at the beginning, years ago, we used to, if we had, say, 10 patients, all that have been diagnosed with, say, a colon cancer, same type histologically, all looks the same, they would all receive the same treatment protocol. That was the standard approach to the treatment of these tumors. But what was identified was that perhaps 2 or 3 patients would actually respond very well to that treatment, but some of them would actually do very poorly and wouldn't respond at all, and others might have a sort of more moderate course. So the bottom line was there was a lot of variability, even though the patient had the same tumor type and was being treated in the same way. And genomics has actually helped us to understand why that is, in that although the tumors may look the same down the microscope, they in fact have different molecular characteristics. And so some may respond better to a particular drug than others. And so we now have to use multiple different drugs depending on the genetic characteristics associated with these tumors. And this is what then underlies the development of clinical trials, where patients will be entered into it. And based on those genetic characteristics, they will be aligned to a certain arm of the trial and a certain drug that they can be started with. So this is how we've actually progressed in terms of our understanding and the way we treat patients. So we can no longer treat patients with the same diagnosis in the same way. What are the genetic changes which underlie disease? Well, I think to look at this, we have to appreciate where we've come forward in terms of our diagnostic strategy with this in pathology. So, as I mentioned, with one key aspect of what I do on a daily basis will be, say, if I got a sample from a patient with a brain tumor, I would look down the microscope, look at the cells, look at the features of them, decide is it definitely a brain tumor? What type of brain tumor do I think it is? But then instead of that being the bottom line, I can now go inside those cells and take out that genetic information and look at the code that might be underlying, and look at the changes that may be underlying that code that may be responsible for the development of that particular tumor. And to go back to the basic principles of what DNA and mutations is, this is what we call the sort of canonical pathway associated with DNA. And to understanding the basics of this is key to understanding what we're looking at when we're doing different forms of genomic testing. So DNA is about 3 billion letters long, and it consists of different orientations of 4 bases known as adenine, guanine, cytosine, and thymine. And within that, the 3 billion letters of code, there are certain regions that have really important functions, and these are what we call our genes. So there are around 22,000 different genes, some of which we know what they do, and others we're still a bit in the dark as to what function they actually have. But ultimately, those that we do know about, they have a really key roles in making different proteins, enzymes, or other important functions that we require to actually exist on a daily basis. And when we look at the structure of genes, there is always a promoter region of a gene, which when this is activated, it helps to switch the gene on and make sure it's actually functional. We have the exonic regions of the gene, which are known as the protein coding regions, which are key bits of code that actually help us to make the protein that we desire. In between these, we have the intron regions, those in red, and these were traditionally viewed as what's known as junk DNA, so didn't really have a particular function at all. But now with a bit more research, we're actually understanding they do actually have an important function to play within this process. Now, with the help of an enzyme known as an RNA polymerase, we then can, and the transcription process, we make a strand of ribonucleic acid or RNA. And this is a key pathway. So almost a mimic of what we got with our DNA strand. But then it needs to then subsequently undergo some processing. And with the help of little proteins known as spliceosomes, the intronic regions are actually cut out of this, and we end up with a strand of messenger RNA. And with the help of a little organelle in cells known as a ribosome, this ribosome will then read the code of the messenger RNA. And according to what code it reads, it will add a particular amino acid to the chain. And the chain gets bigger and bigger until the entire code is read. And this process is known as translation. When that code is then, when that amino acid chain has been made, there are some modifications that take place to this, but ultimately we derive a protein at the end of it. And so that protein then can be used for whatever function is necessary for it. But as with everything, things can go wrong with this process. Now, if you imagine the genome is effectively like a large book, now whether that's a fictional story or a factual textbook, when we're reading our book, reading the particular chapters, reading the paragraphs, we get particular messages of what the author is trying to tell us, and so we can follow it. But as you're reading whatever textbook it might be or book, you may come across particular errors, and it might be a spelling mistake, it might be two words that have been added by mistake, or it may be that the sentence is actually jumbled up and doesn't make sense at all. And so it's difficult for us to derive any meaning of what we're actually reading. And it's the same principle when things go wrong with DNA. And there are a variety of different things that can happen. So it could be that a base is actually substituted into the code. So here we've got a C that's replacing what was a T at this point. You can also get bases that are actually added into the code. So here we've got a cytosine that's been added, and it's caused the code to the right-hand side to completely shift, and so therefore will alter the format of the protein that's actually produced at the end. You can also get bases that are deleted. So here we've lost a thymine at this point, and again we've got a shift in the code the other way. But it may not be just a single base that's added or deleted. It may be entire segments of a particular gene that are either added in and duplicated, or that are actually deleted and lost. But sometimes these can be actually repaired, but other times damage is too much to be repaired, and that is what then leads to the onset of disease. And so these different changes in the sequence are what characterize a number of different tumors and also diseases that we come across on a daily basis across pathology. In terms of when we're looking at cancer, there can be a variety in differences in terms of the number of mutations that or changes within the DNA that we identify. So according to this chart here, you've got a range of different types of tumor that we can get both in adults and children. And here along the y-axis is the number of mutations that can be present within that DNA. Towards this end, you can see some types of pediatric tumors, so low-grade gliomas and high-grade gliomas, that have very few, under 10 changes within the DNA. But up towards the other end of the spectrum, including melanoma and that some types of pediatric high-grade glioma, they can have up to thousands of different mutations within their DNA. And the burden of mutations varies across different cancer types, as you can see, but it also influences the type of treatment that we may want to give a patient. And even though a patient, you may think that one with thousands and thousands of different mutations is going to be, there may not be a treatment available for that. There are some that we can use known as checkpoint inhibitors that some of these patients may benefit from being given. Another change that can happen with DNA is actually something that it happens normally, and that's methylation. And methyl groups are added at various different positions along the DNA chain, and they tend to be where there's a C or a G next to each other in the code, and that's what we call a CPG island. And what we know is that these methyl groups tend to be added at the promoter regions of particular genes. And if a promoter region is methylated, it actually switches the gene expression off. So even in normal DNA, it has a role to play in the activity related to different genes. And the degrees of methylation or not of the DNA is actually influenced by the principle of epigenetics, which is where things in our lifestyle or our environment can actually have an impact on our DNA and what happens to it. And so this is a new sort of field within genomics that we're starting to uncover the function of it and the importance with it related to normal activity, but also related to disease as well. Another change that we can see within our genome are fusions or translocations. And the principles behind this are where you have chromosomes, which are, because our DNA is so big, it's 3 billion letters long, it's all wrapped up and processed into chromosomes so it can actually fit inside the cell. But sometimes, for whatever reason, breakages can happen in the chromosomes. So, for example, there's a break here in chromosome 1 and a break here in chromosome 2. In some instances in genomics, we can get a process called chromothripsis, which is where the entire chromosome shatters. We still don't quite, and then sort of rejoins together in a very abnormal format. And we still don't know quite what happened, why that happens, but a very interesting format that can occur in some diseases. But the problem is that these broken bits can then join to the wrong chromosomes where they shouldn't be. And whatever format that you get related to this, you get two genes that are next to each other that wouldn't normally be next to each other at all. And one of those genes tends to actually cause an activation of the other, and that may be enough to stimulate the development of a cancer. And there are different formats of how these fusions can actually happen. So as I mentioned, you could get the breakages occurring on two different chromosomes, but also just on a chromosome on its own, you may get a segment that's actually deleted from that, and then the two broken bits join together. You may get one that where two breaks occur in the chromosome, the bit comes out, turns itself round and then inserts itself back in, or you may get a segment of the chromosome that's actually duplicated, and so you get two versions of the same segment of the chromosome next to each other as well. But the underlying principle is the same. There are two genes next to each other, and one is causing the overactivation of the other. So having gone through a few of the genetic changes that we can see, what are the actual different molecular tests that we can actually use to try and detect some of these abnormalities that we may find in our genome? Well, if we start with a bit about DNA methylation. So there was a major discovery that was made and published in 2018 in neuropathology. And this was the principle that if you take the DNA from all the known type of brain tumors and look at where the methyl groups are added along the DNA chain, each individual brain tumor will have a different profile of where these methyl groups were added. Now, this was really exciting because frequently each week we come across cases where when we look at them down the microscope or on the screen, we're not really sure what the diagnosis might be because there are a lot of features that overlap with each other. But if we can then sequence where these methyl groups are added, we can then help to support with the diagnosis of what we think it might be. And this led to the development of what we call the Heidelberg Brain Tumor Classifier. So when we get our data of where these methyl groups are added, we can plug that into the classifier and it gives us a result from 0 to 1, 1 meaning that the profile matches exactly with this particular type of tumor, whereas if it's closer to 0, it doesn't really fit within any of the categories within the classifier, and so we need to look for other characteristics of it. And this picture on the right-hand side is what we call a TS&E plot, and it's a way of representing methylation data within the classifier. So each color represents a different tumor type, and each dot within those clusters represents an individual case. And so you can see that tumors that have a similar methylation profile will cluster together in these little clouds. So we can pop our cases into here and see which cloud they actually align with and look at that score to see how closely and reliable that classification actually is. But also what you'll notice is that there is a lot of white space around these clusters as well. And sometimes a case may land in that white space. And that might indicate that we've actually discovered a novel type of brain tumor. And I'll talk to you a little bit later on about some of the examples of where that actually happens. The other important thing that came out from this study was it showed that over 10% of cases that featured within the study had a change to the diagnosis based on the methylation classification that came out from it. Now, some cases actually increased their grade, so they were predicted to therefore be more aggressive than was originally thought based on their histology. And that's important to identify because those patients may need more aggressive treatment in the form of chemotherapy or radiotherapy, or maybe even more aggressive of aggressive surgery. While on the other hand, some tumors were actually downgraded. So the histology predicted that they were actually going to be a very aggressive tumors, but when the methylation profile was derived, it actually predicted they were more low grade. And so we can save those patients from having unnecessary treatments, which can often have significant side effects as well. So a really nice example of how important it is, this form of genomics testing in actually the diagnosis related to brain tumors. And this is the device that actually helps us get all of that information, the bead chip Illumina array. And it's literally the size of a memory stick. And each one of these can process around 8 samples, so 8 different tumors from 8 different patients. And it looks at 850,000 different CPG islands where the methyl groups are added across each one of those DNA strands, and can provide us with that classification. So a very small piece of technology, but provides an awful lot of crucial information. And this has been significantly developed in the brain tumor world, but it's also used in soft tissue tumors and sarcomas. And there is also being developed now a pan tumor classifier where it will feature classifications of all the known types of tumors as well. So it's certainly something that's actually taking the specialty by storm. To show you another example of how important this is and why it's really useful, we did a study back in 2017 which looked at over 1,000 cases of an aggressive type of brain tumor called a high-grade glioma or glioblastoma, usually have a very poor prognosis of around 2 years maximum. And when looking at the histology of these cases, they all showed a very similar picture down the microscope. So they could all be diagnosed as the same type of tumor. But when looking at the methylation profiles and plotting them into our TS&E reference cohort of these different types, I'll show you where they all landed. And you can see there is a spectrum of them landing into all of these different colored subgroups. And so now in research, we have to identify what the different genomic characteristics and mutations within that DNA that are actually underlying those individual cases. And so it presents us with a really interesting profile now where we have different subtypes of tumors of similar type that actually are characterized by different genetic alterations. But there is still a significant problem because all of these gray ones here represent those tumors that we still don't really know an awful lot about and what is actually driving them genetically. But each year, this gray bar gets narrower and narrower and narrower. And this is the same across other different tumor types as well, not just in brain tumors. And what's driving that change and that understanding? It's the greater use of genomics within research, but also in clinical practice as well. So we're helping to demystify some of these unknown characteristics of tumors. Fluorescence in situ hybridization is another test that we can use, and this can help us to identify increased numbers of copies of particular genes or changes in chromosomes. So the principle behind this is that we label the genes that we're interested in with a fluorescent marker. So here we've labeled the one we want in red, and we also label a control gene in green so that we've got a control to compare to. And so normally in a normal cell, you would expect perhaps 2 copies of the same gene in each cell. But in the one on the right, which is a cancer cell, it shows there is a vastly increased number of copies of this particular gene. And in this tumor, it was a gene known as MYC-N. And this is amplified in a pediatric type of tumor known as a medulloblastoma, and is important to identify because it tends to show that this tumor is much more aggressive. And so therefore the oncologists need to treat it more aggressively as well to try and get control of it. So a useful test in helping to identify and prognosticate for certain tumor types. But it also can be very useful in identifying fusions as well. So if you suspect a particular type of fusion within a cancer, you can label your genes of interest again. So in this case, we've labeled ABL gene in red, which is on chromosome 9, and we've labeled the BCR gene on chromosome 22 in green. And so in our normal cell, again, you would expect just 2 copies of each of the genes. But in these tumor cells, what we can see is that the green and the red signals have joined together. Normally, they should be far apart because they're on different chromosomes, but they've joined and giving a yellow signal indicating the presence of this BCR-ABL gene fusion, which characterizes a type of leukemia known as chronic myeloid leukemia. So very useful in terms of helping to diagnose these tumors. DNA sequencing is something you will hear an awful lot about nowadays, and there are a variety of different forms of sequencing that we do. And when we say sequencing, what we're doing is actually reading the code of the DNA strand. And so whole genome sequencing is where we're basically reading the entire strand of the DNA. And that one takes a long time because there's 3 billion letters to go through, but also it's very expensive and very time consuming in terms of the data analysis as well, and requires an awful lot of storage space on computers as well. So we don't necessarily do it on all patients, but in some tumors where it's perhaps a novel one or we don't know a lot about it, it can provide very useful information about the background genomics. The other thing we can do is hold exome sequencing, where if you remember those exonic regions of particular genes, we can just sequence those protein coding regions. And that may be enough to tell us the genetic information or the abnormalities that have gone wrong with that. And in those tumors that we know a lot about, we can refine it even further. So we can do what we call panel sequencing. So we may choose to only sequence a few select genes that we know are the ones which can be abnormal in certain types of tumor. So it could be just a panel of 5 genes. It could be 10, 20, 50, 100, 300, depending on what number of genes that we know are abnormal in these particular cases. And that goes the same for different diseases, not just tumors. And the principle behind the sequencing is what we do is we fragment the DNA into small fragments and pieces, and then we add adapters to the ends of those DNA fragments. And there is a different adapter for each patient. So when we're sequencing, it means that we can sequence multiple patient samples together and to derive the data, helping to generate the data quickly so that we can get the treatment started. And we replicate or develop more of these strands and fragments through processes of PCR, polymerase chain reaction, and then using little fluorophores that bind to the respective bases on these DNA fragments. And when they bind to it, they let off a light signal that's detected by the sequencer. And depending on what signal is detected, that corresponds to the particular base that has been detected. And then our data analysts will compare the code that we derive from the sequencing to our reference genome code of what it should be normally. And we line up these different sequences. And if there is an abnormality within it, we can check it and see that it's different to our reference cohort, hence identifying the abnormal mutation. And it may be just a single gene or it may be multiple genes that are affected. And so what we do is we keep a close eye on the number of these different sequences, or what we call reads, that contain the different mutations, because that can be quite significant in terms of the outcome for patients. So now a bit about how genomics can actually help with research as well. And just to introduce to you a little bit of work I did related to my PhD. So I was interested in looking at high-grade gliomas, those aggressive brain tumors that occur in children under the age of 4, so very young children. They can occur in any location within the central nervous system, including the brain, cerebellum, the spinal cord. But it had been known for a while that although they've got a very poor prognosis, they do slightly better compared to other high-grade glioma types. And when I gathered tumors from our colleagues from around the world and did some methylation profiling of them, what I found is that these tumors landed in a space separate from all the other known type of brain tumors, which suggested that they were in fact a novel or new type of brain tumor. So I wanted to find out a little bit more about them. And so what I did was did a bit more genomics testing. When I looked at them down the microscope, they showed the typical features of a high-grade glioma, so very aggressive. All of this here is showing necrosis. There are lots of tumor cells here, which again is all a characteristic feature of an aggressive type of brain tumor. But no surprise that they were graded as a high-grade type tumor. But when I looked at this clinical data for them and the survival data, these children were actually doing a lot better. And so they were surviving despite the fact that the histology predicted a very poor outcome. And when I turned to genomics, I managed to get my answer. Because when I did some sequencing, so I did some whole genome sequencing and I did some whole exome sequencing to start with, I didn't find any changes in the DNA at all. There were no abnormalities that I picked up on it. But when I did a special type of sequencing that looked for the presence of fusions, I identified either just one of 4 different genes that that could contain a fusion. So ALK, NTRK, ROS1, or MET. In each one of those tumors, one of those genes was involved in a fusion. And there were lots of different partner genes that were responsible for the overactivation of those genes. But it seemed to be that that was what was driving the development of this tumor. Now, in research, one of the other things that we're privileged to be able to do is when a patient with a tumor that we're particularly interested in studying is in theatre, we can attend theatre and collect a small sample of that tissue, put it into some culture media, rush it back to the lab, keeping those cells alive, and then grow them into culture flasks that we can then perform different tests on. And that includes testing them for their susceptibilities to different drugs. And so during my PhD, I was able to get hold of 2 cases from patients who had both of these tumors had an NTRK fusions within them. And when I tested them with the 3 drugs, entrectinib, crizotinib, and milsiclib, all of which target this particular fusion, they all showed sensitivity to the drug, which meant that as the concentration of the drug increased, the tumor cells died as a result of it, which meant this was a really exciting opportunity and possibility for the treatment of these patients. Now, what about if this is translated into patients? Well, it was. So these patients, if you remember, were diagnosed with a high-grade aggressive tumor with a 2-year prognosis. Many were then started on these treatments and they survived. They did remarkably well and are well and alive today without any disease present. And so this actually led to the development of clinical trials for these patients and the inclusion of this new type of brain tumor within the WHO classification of brain tumors. So an exciting step forward in the classification of tumors. And I'll show you at the end of this talk a particular case example for one of these patients. Another bit of work that I very recently published last month also shows the value in genomics in terms of our spectrum of what types of tumors occur in both adults and children. So if you look at this picture on the right-hand side, this is an age density plot. So you can see the age along the bottom axis here, so increasing age as we go along. And if we look at the top 5 boxes here, you can see that these all have peaks associated with patients under the age of 25. But what's very interesting is there are also peaks in older patients as well. So even those in their age 75 will also develop one of these tumors potentially. And what's interesting about these groups is that they are pediatric type tumors. And so genomics is really important because if we just rely on histology alone, we wouldn't be able to pick up these pediatric type tumors occurring in adults, older adults. And it represents a really interesting opportunity for perhaps studying these in more detail and understanding what characterizes them and potentially using other treatments for them as well. Research is also moving on significantly now because instead of just sequencing all the DNA from a tumor at once, we can also now take out the RNA from single cells within the tumor and look at the particular characteristics of it. And so this allows us to explore what we call the heterogeneity of the tumor, looking at the different populations of tumor cells, but also looking at the populations of immune cells that may be present within that and other cells in what we call a tumor microenvironment to see how they might interact with each other. And leading on from this, with something that I'm very excited by, is that we now have the potential to actually label on slides where these cells are with particular genetic alterations. And this allows us to map these different genetic characteristics of tumors and then identify where the ones are and what their significance may be in terms of behavior. So where the tumor is infiltrating, we may be able to identify its particular changes in the DNA at this point and highlight that that may be an important change to target in the future. One of the big challenges in terms of genomics is the turnaround times of our results. So it can take between 2 to 3 weeks or sometimes months to get the results back of these tests. And all the while, the patients are waiting for their treatment. There's a revolutionary technique now known as nanopore sequencing, which is being trialed at various hospitals around the country at the moment, which is potentially giving us the same genomic information, but within a matter of hours. So we may find that patients who are in theatre having an operation, by the time they wake up from their anaesthetic, we will have the full genetic workup of these particular tumours, which is exciting in terms of starting their treatment promptly. Something to mention briefly that you may have heard of is CRISPR-Cas9 technology. There isn't time to go into the specifics of details of this, but effectively these are molecular scissors which can actually cut segments of DNA out. And it allows us to actually understand the function of particular mutations in research. So if we identify a mutation in a particular tumor, we can cut the DNA, cut that change out and join the DNA back together and see if that tumor cell actually becomes a normal cell again. Alternatively, we can actually, in normal cells, insert by cutting and then inserting a mutation into the DNA to see if that tumor actually transforms into a tumor cell. This has the potential also for being used in clinical settings for treatment of patients. And in certain diseases like sickle cell anemia or thalassemia, this form of technology is now being utilized with great effect as well. I've talked an awful lot about neuropathology and histopathology in the talk tonight, but all the other specialties in pathology also use genomics to various different extents as well. And that's hematology, chemical pathology, virology, microbiology, veterinary pathology, and immunology, to name some of them. So all of them have the option of using genomics to make a diagnosis. So whichever specialty you might choose to get involved in, you will find genomics as part of that clinical practice. So just to finish, I just wanted to show you a few quick case examples where genomics can actually help patients. And the first one is a little bit related to the research I showed you earlier on in my PhD. So this was a baby that was was diagnosed with a brain tumor before they'd even been born. Shortly after birth, they had a biopsy that was diagnosed as a high-grade glioma, so predicted to have a very poor outcome with this. The little baby, only a few weeks old, underwent surgery to have the tumor removed, and a sample was sent to us to have a look at. Histologically, again, it showed all the features typical of an aggressive high-grade tumor, so again, predicted to do very badly. But when I did the investigations for this, what I found was that there was a break at chromosome 12 at this point and a break at chromosome 15 at this point, and the two broken bits are joined together, leading to a fusion between the ETB6 and the NTRK3 gene. This was confirmed with fluorescence in situ hybridization. Again, you can see the ETB6 NTRK3 labeled in green and red, and we've got the joining of those two signals indicating the presence of the fusion. But what is the significance of this for our patient? Well, this is the tumor that was the MRI image of the tumor before the surgery. And then despite the best efforts of the surgeons, they couldn't resect all of the tumor. So this is a highlight. The residual tumor is highlighted in red here. And then based on those results of the genomic testing, this patient was started on crizotinib, and I'm delighted to say that she is now 7 years old and the tumor has completely disappeared. And when you think about this being a high-grade tumor, that's a remarkable outcome. And it's not just this patient, but there are many others in a similar way now as well. The next patient, this is a 49-year-old male with a history of smoking, chronic cough, blood-tinged sputum, and has been losing weight. So we're quite concerned about this history. And the imaging suggests that there is, in fact, very suspicious for a tumor here. And that's, in fact, what has been identified. The patient undergoes a bronchoscopy with some bronchial washings, which are sent to us, and also a biopsy is organized as well. And in the bronchial washings, you can see the normal type of respiratory epithelial cells with their cilia on the surface and a macrophage here. But what we see here in our patient sample are clumps of very atypical looking cells. So again, suspicious that this is a tumor. The normal histology of the lung, here you can see the alveolar air sacs, and in our patient, this is very different. So these are horrible glandular tumor cells, all lining up with this mucus-like material in the center. And this is typical for a lung adenocarcinoma. Years ago, that would be the end of the story. But now with genomics, we can do further testing. And by doing fluorescence in situ hybridization, looking at the ROS1 gene, we identified a fusion, which this patient is targetable. So this patient could then be started on a targetable treatment. And the final case is a 28-year-old with a very worrying skin lesion, which we're suspicious might be a melanotic lesion. So the patient has a biopsy, and it's where we take an excision biopsy, where we give a nice boundary of normal skin to ensure that we get clear margins associated with it. Down the microscope, it looks very nasty, so very atypical looking cells, and this brown pigmentation, which is typical for a malignant melanoma, which is in fact what this was diagnosed as. So that would normally have a very poor prognosis with it, but DNA sequencing has actually helped in this case. And by sequencing a particular gene known as the BRAF gene, just a single change of a T to an A meant there's a change from valine, the amino acid valine, to glutamic acid. And also, in some cases, you can also see with the changes of two bases here from GT to AA, either one of those means that we've got a BRAF mutation within this tumor, and that is also targetable. So this patient could be started on some treatment to help them respond to that. So in summary, genomics is now an integral part of diagnostic practice. It helps us to identify the mechanisms underlying particular diseases. It helps us, the research that's done in genomics has allowed us to make significant advances, not just in brain tumors, but across all cancer types, including the use of targeted therapies. The genomics results are required now for actually entry into clinical trials. That's how integrated it actually is. But the costs of genomic testing remain a challenge, and we need to think very carefully about that for lower middle income countries and ensuring everybody has equitable access to these tests. So it's really transforming pathology and helping us make really precise diagnoses and risk prediction in a wide range of diseases, and really helping us to integrate genomic information and effective care, improving outcomes for our patients as well. And just as a final slide, this is a really nice picture it's based around the Andy Warhol soup cans artwork. And this was put together by my boss, my supervisor, Chris Jones at the ICR. And what this nicely demonstrates is we all know what soup is. These are all cans of tomato soup, but each one of these cans is a slightly different flavor and it's got a slightly different color. And I think this is what we typically see with disease now and illustrates quite nicely what genomics has done. We need to, instead of just looking from the outside, we need to go inside now and look at those different ingredients to understand more about what's actually underlying that. And that is what genomics is doing for us and for our patients as well. So these are just some of my contact details. I'm very happy to be contacted outside of this webinar if anybody's got any questions about what I've talked about or about careers in pathology if they're interested in as well. I'm very happy to take any questions that you may have tonight as well. Thank you very much for listening.
Thank you for your talk, Dr. Clark. That was very insightful and engaging and super interesting. So we've got a few questions on the chat already. If you haven't put them in yet and you have something to ask, just type them up. So someone's asking, where would genetic counselors come into the diagram of people involved in this?
That's a really good question. So genetic counselors are a vital part of the work we do as well. And as I mentioned, there are several people that haven't been mentioned on that there's a complex web and network of people involved in that. So it may be, particularly when we're suspicious that there may be a particular genetic alteration that's maybe affecting family members or have implications to that patient, that it's even genetic counselors get involved even before the testing is actually done. So may counsel them about the results that we may expect to find associated with particular diseases, but also they have a role then throughout the whole process. So then when the results come back, what they actually show and what they mean, both for the patient, but also for subsequent generations of the family as well. So really knowledgeable and important people in that whole process as well.
Okay. And then the next question says, how significant of a contribution might artificial intelligence have in its role in the diagnostic side of pathology? Are specialties such as histopathology vulnerable to being replaced or made less important?
Yeah, no, that's a really good question and something that is a fear across the whole community. And I think that's something we see with change generally. We all fear that it's going to replace us. This was a fear associated with genomics and molecular pathology. And look, it hasn't done that at all. It's actually now a tool in the toolkit of pathologists to actually help us improve our daily practice. And it's the same thing with artificial intelligence. So, for example, artificial intelligence may be useful in helping us to identify or process genomics data. It may help us to be able to look for patterns across a wide network of different genomics data across different patient sets that we hadn't seen. And it's these patterns and commonalities which we may miss that actually could be really helpful in identifying key characteristics of particular tumor types as well. We can also use it in pathology, certainly to count the number of percentage of certain cells with particular characteristics are really important in certain tumors. And we could, rather than relying on me counting them individually, I'd much rather rely on a reliable program that can do that for me much more accurately. So I think there's a lot of different avenues where artificial intelligence is going to help us, and yeah, looking forward to seeing how that will develop in terms of the genomic side as well.
Okay, great. The next question says, what specific technology did you use for sequencing, looking for fusions for the infant gliomas?
Very good question. Yes. So this again, when I came into this, I had very little idea about genomics or what was going on this. So this was a massive learning curve for me. What we ended up doing was we looked in the literature at the number of there was a key case that we picked up one of these NTRK fusions by doing some whole genome sequencing and confirming that with fluorescence in situ hybridization. So we felt this was a hypothesis that they may be occurring more frequently in this group of tumors. So we designed a panel looking, I think there were about 30 genes as part of it that we knew were associated with translocations and fusions in brain tumors. And so this was a DNA specific panel. And so we ran that and sequenced those and looked for particular breaks and the joining together of these particular genes to identify that. Often to confirm the presence of a fusion, it's nice to do two tests that help to validate each other. So you could do fluorescence in situ hybridization, but the other thing you can do is RNA sequencing, because if the RNA shows the expression of the fusion, it shows that it's actually valid and more genuine. We can get lots of breaks in the DNA that can happen anyway, but they may not necessarily result in a fusion. So there were a couple of techniques that we used to actually help confirm the presence of those.
Thank you. The next question says, do you have any tips for getting work experience in genomics before applying for jobs after graduating?
Yeah, good question. I think there should be plenty of opportunities, and I think my key starting point would be to discuss with your local pathology department, because as I've shown, again, it doesn't matter what specialty, and you may have a particular interest in a particular area of genomics, maybe that you want to explore more about, but having a chat with them about that might be a useful opportunity to then perhaps do some shadowing within the lab and see how the tissue is actually processed and actually prepared for genomic testing. The other thing is to consider the research institutes. So some of their practice on a daily basis is going to be genomics related as well. So arranging some work experience in one of those labs where you can perhaps shadow some of the researchers as they process some of the donated samples might be another strategy that you could use. But either one, you should be able to get some exposure and some understanding of what goes on in the lab system to help you.
Great, thank you. The next question is there a risk of losing detail when increasing the speed of genomic testing when using nanopore technology?
That's a really good question, Jessica. Thank you for highlighting it. I think what one of the big challenges, it's still a relatively new technology, but what we're doing and what we're deriving and what we're testing is to see how reliable, what level of sequencing we need to do to get a reliable classification and a reliable level of mutations from it. So that's what these studies and these trials are aiming to do. So at the moment, we're doing the nanopore sequencing, but then comparing that to our established techniques of doing the methylation profiling or the other sequencing, and then comparing the results to see how accurate the two are and whether they match up or not. One of the things we're hoping to do is within the hours, be able to give a reliable classification by methylation, and that will be able to tell us, is it a high-grade tumor? Is it a low-grade tumor? Is there anything that's particularly, what type of tumor it might be, whether it's a glioma, an embryonal tumor, or a glioneuronal tumor? All of those will have different formats of treatment that the oncologist will want to start as soon as possible. And so if we can give them that information at that stage, we can let the sequencing carry on for a lot longer, then get more detailed information, perhaps even whole genome data that we can then utilize at a later date for it. But I think the key thing is to try and get as much information to help with a quick diagnosis, and then we can carry on doing our usual investigations after that as well.
Thank you. And then next question says, thank you for your talk. It was very interesting. In your PhD publication, you mentioned how this work led to use of NTRK inhibitors for children with NTRK fusion. Have there been any advances in targeting the other fusions you mentioned, e.g. ROS1, MET?
Thanks very much, Ruben. You're really nice that you picked up on that. Yes, so the examples that I showed were related to NTRK, but actually in the paper as well, there were some examples where ALK has been targeted as well. But what's really nice is all these genes, whether it's ALK, ROS1, MET, or NTRK, all come under the heading of tyrosine kinase receptors genes related to that. And so interestingly, they use drugs that target the same thing in all of those. So whether it has a ROS1 fusion or a MET fusion, you can still use those drugs to actually target that particular change. So that's really, really helpful. One of the exciting things about these fusions is we've known about them for a long, long time. So we've known about NTRK fusions in, for example, colorectal cancers going back to the 1980s, but it's only in recent times where we've actually seen that crossover where they're actually occurring in these very young children. Interestingly, in the studies and research we've done, we don't see them nearly as frequently in the older adult population. And so we've still got to do a bit of work really to understand why in very, very young children these fusions tend to occur at this stage. But yes, those we can treat them in exactly the same way with the same drugs at this stage. The other thing with them, so there are some fusions that occur in slightly older children, but often they're not just isolated, unlike in the infant. So there may be other alterations that can occur in the DNA in the background. And so there may be a response to that in terms of the treatment, but there are other things that are still going to carry on driving the tumor as well, which may require other treatments. And so we still got a lot of work to do to elucidate and identify better treatments for those patients as well.
Okay, the next question says, how much of a delay from presentation to effective treatment commencing does genetic panel screening take? Would some cases benefit from the previous, I was meant to say, i.e., what is the trade-off between diagnostic delay and improved outcomes from tailored therapy?
Really good question. So in relation to the delays in treatment, so as soon as I get a sample that comes through from a patient, I get my clinical information of what the background is. I have a look at it down the microscope or on my digital screen in front of me, and I can make an assessment of what type of tumor I think it is. And based on that, I can then order those molecular investigations immediately, so they can then start being processed. And so we want to expedite the use of the tissue as effectively as possible. In my assessment, I'm also trying to keep in my mind what investigations I'm going to want to do, because often we receive literally tiny pieces of tissue. So it may be only a couple of pieces of tissue, only a couple of millimeters in size. And so I need to balance what is the best use of that tissue. Is it perhaps more molecular investigations, or is it other things that I can do in the background as well? Usually it takes around 2 to 3 weeks to get those results back. And we can give the oncologists and the surgeons a preliminary diagnosis of what we think we're dealing with, but we have to wait a bit longer to actually get those genetic results back. So it's all done as quickly as we possibly can, but these latest advances in terms of speeding things up will hopefully help to remove that delay and ensure that we can start treatment a bit quicker. Because again, we don't want to be hanging around and giving any unnecessary delays with it. But unfortunately, there's nothing we can do particularly that can expedite it any quicker at the moment.
Thanks. We've got 2 more questions, so maybe, three now. So maybe those will be the last few, and then if anyone has any others, just email Dr. Clark, just in the interests of time. So the next question is, how is the work between the genomics and the pharmaceutical side coordinated to ensure that as the new genetic profiles are detected, there are treatment options for them?
Thanks, Eleanor. Another really good question. So this is where research within genomics is really key with this. So in the research team that I'm part of, we have within our team themselves, we have experts who will actually screen our samples in terms of their drug sensitivity. So we can do really large screens of our tumor cells with hundreds of different molecules to assess any sensitivities of those. And then we can pick out those little molecules to actually decide whether they're worth pursuing any further with that. And within research institutes as well, there are drug discovery teams that have huge experience and backgrounds in chemistry and medicinal chemistry, etc., in designing drugs related to this. So there is a big link between the research community, pathologists, and the clinical teams. And it's important to keep that link going and feeding back that information. And presenting data in research meetings with the clinical teams involved really helps to actually ensure that information is shared on a frequent basis to keep everybody updated about that progress. It takes quite a lot of time to develop a drug from very small molecules into something that's available in clinical practice, but that doesn't mean we shouldn't still be pursuing it, particularly with high-grade tumors. And so some of these genetic alterations that come out of research may highlight to these drug discovery teams that this may be a focus for future work, particularly if this is an alteration that's shared across many different tumor types as well.
Thank you very much. So that was the last question. If your question didn't get answered, sorry about that. Please just send an email to Dr. Clark.
I'm very happy to. I will answer those outside of the meeting, Zainab. We'll make a note of those, so that's no problem.
Okay, great. I'm just going to hand over to Professor Davies now.
Okay, well, thank you very much, Matt, for a fantastic presentation. If you'd like to read more about this topic, Matt actually co-authored a really great article on it for January's issue of the RC Path Bulletin, which you can still find on the RC Path website if you want to take a look. Huge thanks as well to Rosie and Zainab for all their help with presenting this webinar, and to all of you for your fabulous questions, which I was really impressed with. We'll send you a short feedback survey by email, so please share your thoughts with us about this event on that survey. You can watch our previous careers webinar on our YouTube channel. We'll share the link with that in the email, or you can scan this QR code here. We'll be adding the recording of this event to the playlist, and our next webinar is about paediatric and perinatal pathology, and that will be on the 19th of June at 6 p.m. So we hope to see some of you there as well. And finally, you can find out more again about pathology on our YouTube channel and our Instagram, which are on the slide there. So it's now time to close this webinar. Thank you all very much for attending. I hope that you found it useful, and have a lovely evening, everyone. Bye.